You're still copy-pasting, we can maybe continue with our first lecture, which is about preprocessing of EEG, MEG datasets and channel level analysis. And the outline of this talk is as follows. So first we will have a look at what kinds of signals are actually generated in the brain. Afterwards we will look at how we can record these signals with electro and magnetoencephalography. Then I will briefly introduce to you the example dataset that we will also use during the hands-on sessions, which is a somatosensory experiment. And the main part of the talk is how we can use FieldTrip to do the pre-processing of these EEG, MEG datasets. So I will present to you different functions that FieldTrip offers. And in the end, there will be a short summary. So now we will have a look at what kinds of signals are generated in the brain. So if a neuron is active, it fires an action potential, which is mainly a pulse of approximately 100 millivolts that travels along the axon of this activated neuron. And this action potential comprises a depolarization phase and a slightly longer repolarization phase. And after this action potential, the neuron sends neurotransmitters, which bind to the receptors of the following, the postsynaptic neuron. And this postsynaptic neuron then gets either depolarized or hyperpolarized, which is an excitatory or an inhibitory postsynaptic potential. And these potentials summit, and if a certain depolarization threshold is reached, then an action potential is again generated. There's a short sketch of what's happening there. So in order to be able to measure the electromagnetic activity outside of the brain, it must be of sufficient strength and duration. And a typical action potential lasts around 1 to 2 milliseconds. So it's very short, while the postsynaptic potential is more than 10 milliseconds usually. So it's way longer. So we can say that EEG, MEG mainly, the signals originate from a synchronous activity of a large number of neurons. And the signals mainly stem from pyramidal cells in the cortex, which are parallel and oriented perpendicular to the surface. And EEG, MEG doesn't measure postsynaptic potentials, but just measures postsynaptic potentials, not action potentials, because they are too short and not synchronized. Here you can see a microscopy image of a typical pyramidal cell layer in the cortex of a primate. Now we will have a look at how we can measure these signals with EEG and MEG. So first we will have a look at EEG. You usually have a standard set of electrodes like the 1020 or the 2020 system you can see on the left, and then you attach these electrodes to the head. You add some reference channels. You measure the impedance of each channel, and then you record the head surface the signal relative to some reference channel. So you have something like this that you can observe for each channel a signal. Now if we turn to MEG, electricity is always accompanied by magnetism, so every dipole is also surrounded by a magnetic field, which is perpendicular to the current according to the right-hand rule. So as you can see here, we have a tangential dipole. Tangential means that it's parallel to the surface. Now, the main challenge in recording these measurements from the brain and MEG is that the signal that the brain produces is really small. So we can see here that a typical evoked response from the brain is around 100 femtotesla, and there are even larger signals coming from the heart or muscles. Then we have a lot of noise in the environment. The Earth's static field is huge compared to this response, and as a comparison, a typical MRI uses signals in the range of Teslas. So we have to measure this tiny signal that the brain produces. And there are different ways to deal with this. So one is that we have very sensitive magnetic detectors, which are called superconducting quantum interference devices, or SQUIDs, which is a very sophisticated technology, and basically they enhance the signal by superconducting materials, and they need to be cooled down by liquid helium, which also makes the MEG quite expensive. Another way of dealing with this is to handle the environmental noise. So for example, we use a highly shielded room. This is our CTF MEG machine at the IBB in Münster, for example, which has a very many layers of isolation in this room. Then you can also use reference sensors for detecting noise and subtract it from the signal, or you can also use different sensor designs to deal with noise. In MEG, there are three main types of coils. So you can either have a magnetometer, which is a single coil. You can have axial gradiometers, which are radial to the surface of the head, or planar gradiometers, which are tangential. That mainly depends on the system that you have. So today we will use a CTF MEG, which mainly comprises axial gradiometers. And for the same magnetic field that you can see in green here, they produce a different output, so they're differently sensitive. So for example, the magnetometer and the axial gradiometers, they have a peak and a sink, while the planar gradiometers is symmetric. So a typical answer for the auditory evoked responses, for example, for axial gradiometers is depicted on the left, where for each dipole you have one maximum and one minimum located next to it, while for the planar gradiometers you only have peaks directly above the dipoles. So you have to take this into account when analyzing your data. Now I will briefly compare EEG with MEG. So although they measure the same signals, or they stem from the same activity, they have some different features. So the main advantages of EEG are that it's relatively inexpensive. You can port it relatively easy, and it can detect both tangential and radial sources. While on the other hand, MEG is quite expensive so far, and it's relatively immobile because you have this shielded room and this drawer. In the future, this might change. There are some new technologies emerging, like optically pumped magnetometers, so maybe there are some advancements in the future. And another shortcoming of MEG is that you can mainly see the tangential component of dipoles, and you have to it has been shown. Now, just very briefly, to have an overview, I will compare EEG MEG to other methods used in cognitive neuroscience. So the main advantages of both methods are that they are non-invasive. You just measure the signal that the brain generates. They have a highly temporal resolution, which is also quite unique. And they directly reflect neuronal activity, as opposed to, for example, fMRI, where you only look at the oxygen levels. However, they have a relatively low spatial resolution, and it's also harder to detect deep sources because, of course, the sensors are located further away from deep sources, so it's harder to get a good resolution there. Now, I will briefly present to you the example dataset which we use, which is a somatosensory stimulation. So just very briefly, the somatosensory system is about the perception of touch, and it comprises the primary somatosensory cortex, which is located in the postcentral gyrus. And it consists of Brodmann areas 3, 1, and 2 mainly. And I'm sure you've seen this as sensory homunculus. So every body part is represented in a certain area in this cortex. And the secondary somatosensory cortex is located a bit more to the lateral side in Brodmann areas 40 and 43. Now, in the experiment we had the median nerve stimulated at the wrist, and we used the lowest strength that achieves the twitching of the thumb. And the advantage of this experiment is that you get a very large and robust signal, especially early components, but of course you also produce some sort of artifact with this measure. So what we did, we have a right-handed participant. We also measured MRI images, T1 weighted. We have an 80-channel EEG and a 275-channel CTF MEG system with axial gradiometers. Then we stimulated the wrist, 200 milliseconds with varying interstimulus intervals. Now I will come to the main part, so how we can preprocess the data with FieldTrip. And the preprocessing pipeline mainly consists of the following steps. So first we're reading raw data, then we sometimes need to filter the data, we need to remove artifacts from it, and then we average over several trials, and then depending on what you're doing, you continue. So I will start with how you set up the preprocessing pipeline. So usually we study brain responses that follow the presentation of a stimulus or a cognitive event. So you have some event and then you measure what the brain is doing. And then you repeat that multiple times and then you average these signals, like you can see here. And it's very important to know when this event really occurs. So in order to communicate between the stimulus and the measurement, you need to have some sort of feedback, which is what we call triggers. So these triggers in the dataset allows us to identify in the data when the stimulus was actually applied. Now, trials are pieces of data that are used for preprocessing. They're usually a fixed time before and after the trigger, and the trigger is usually T0. And if you define your trials before preprocessing, you save a lot of resources. So if you have a very large interstimulus interval, but you're only interested in the first components, then you can just cut the data before you do any additional steps, just to avoid many recalculations. Now, I hope you're all familiar with MATLAB. So this is how a general FieldTrip function looks like. So you usually have this configuration structure, which you first set empty just to ensure that it's not already set in some earlier function. Then you pass these key value pairs in the way you can see here. So just with cfg.key1 equals value 1 and so on. This is really depending on the function that you have. And then you call the function with this configuration struct, some data input and some optional additional parameters. And then you get this data out format. So if we look at the first step, we can use in FieldTrip the function ftDefineTrial and ftRedefineTrial, where you can define the triggers and the pre and post stimulus interval that you want to use. So this is an example how it could look like. So now we're interested in the first 100 milliseconds before the trigger and the 200 milliseconds after the trigger. We're only interested in the right arm stimulation. And then we call this function ftDefineTrial. Then there's also one other important function, which is called ftPreprocessing, where you can select channels, do a baseline correction, for example, or filter, which we will also see later. Now, after this, a typical structure in FieldTrip can look like this. So, for example, you have information about the electrodes, you have a sampling rates and sample information, the number of trials and timing, and the labels of the electrodes, for example. Now, the next step is usually the filtering of the data. So, the aim of this is, of course, to reduce the noise that you have and to improve the quality of the data. So two typical types of filters are the notch filter, which you can, for example, use to filter out the power line noise, which is usually at 50 or at 60 Hertz and the corresponding harmonics. Or you can use also bandpass filter or low pass and a high pass filter to reduce the frequency spectrum to your range of interest. So this can be, for example, guided by literature, or you're interested in alpha waves, so you have some interest, and then you can only look for these frequencies in the data. So how do we do this in FieldTrip? There are mainly the features DFT filter for the notch filter, and you can pass the frequency with DFT frag. And for the low pass and the high pass filter, we have HP filter and LP filter and HP frag and LP frag. But you will also see this later in the hands-on. Now, how we do artifact reduction with FieldTrip. So typical artifacts that we observe in the data are, for example, the cardiac artifacts from the heart. Then we have ocular artifacts from eyes, and as already mentioned, we have some power line noise. So how do we deal with these noises? So two main ways are the visual inspection, so you just scan the data and look for outliers and manually remove them. You can also do an independent component analysis, an ICA, where you estimate independent components and reject the ones stemming from the artifacts. And we will mainly focus on visual inspection here. So you have this function FT reject visual in FieldTrip. And for example, you can pass the method trials, so you can scan through all trials and just look if some are really messed up and just delete them from the data. Another option is to use the method summary, where you can have different sorts of metrics. In general, that opens a window with a nice graphical interface, so you can just click on outliers in the data. So, for example, if you see some dots there that are really off, depending on your metric that you use, you can just delete them graphically. Now, the last point is the averaging. So, usually we have several trials, this trial itself might not give you the information that you want, so we need a lot of them, and then we average over each trial to get a signal like this. So this is how it could look like, and the more trials you have, the more clear the signal will be in the end that you have. So you can identify components usually only after you average over several trials. This also leaves out a lot of noise that you produce. Another way to look at the data is also to see the global mean field power, which is depicted here. This is mainly also a measure of the amplitude or the power of the data. This is another way you can visualize your data. And in FieldTrip, we mainly use the function FT time log analysis to average the dataset. And in order to compute the global mean field power, we have the option FT global mean field. Now, in the end, I have a short summary. So we've seen that EEG and MEG is a non-invasive way to record brain signals with a very good temporal resolution compared to other methods. EEG measures the electric potential at the scalp, and MEG measures the magnetic field around the head. These signals mainly stem from postsynaptic potentials of the pyramidal cells in the cortex. I've briefly introduced to you the example dataset with the somatosensory stimulation, and we've looked at different FieldTrip methods that you can use to preprocess your data. So, for example, we talked about FT-defined trial, FT preprocessing, and so on. Now, I'm sure we can answer a lot of questions in the hands-on, but are there any questions so far?